Welcome to the Innate Immunity module, where we're going to learn about the alternative pathway of complement activation. In this talk, we will focus on the different ways by which the alternative pathway is activated, as well as what are the outcomes when the alternative pathway gets activated. The complement proteins are a collection of more than 30 different soluble proteins that are present in blood as well as bodily fluids. They were initially discovered in vertebrates. However, now we know that homologs of some complement proteins are also found in invertebrate organisms. A lot of complement proteins are Thioester proteins, and when we look at vertebrates, the complement proteins are mainly synthesized in the liver. Now, the complement proteins work together and form a cascade that's called as the complement pathway. The complement pathway aids in the pathogen elimination as well as induction of inflammation. These complement proteins on their own are not antimicrobial in nature, but when the complement pathway gets activated, that is when pathogens can get eliminated. Now, when a pathogen is able to overcome the epithelial barriers of the host and enter the host, that is when the complement system is activated. There are three main pathways of complement activation that have been studied in vertebrates. The first one is called as the alternative pathway. The second one is the lectin pathway. And then we have the classical pathway. Now, the complement system is an ancient form of host defense, and so when we look at the evolution of the complement pathways, the alternative pathway evolved first, followed by the lectin pathway, and then the classical pathway. The alternative pathway, which is an ancient pathway, can be activated by two ways. The first way is through the activation of the classical or the lectin pathway. And the second way, which is the ancient aspect of the alternative pathway, is the spontaneous pathway. Now, the spontaneous pathway is what we observe in lower organisms as well. But let us first look at the activation of the alternative pathway that is mediated through the classical or the lectin pathway. During the activation of the classical or the lectin pathway, we have the production of C3b. C3b is a fragment that is generated from the complement protein C3 through the action of an enzyme called C3 convertase. This C3b that is formed normally covalently attaches to the surface of the pathogen. And when it is formed by the classical or the lectin pathway, this C3b can bind to another complement protein called factor B. Now, the C3b that is bound to factor B can now interact with another complement protein called factor D. Factor D will then cleave factor B into two fragments, which are called BA and BB. The way we denote BB is it's written as uppercase B and lowercase b. This capital B small b fragment of factor B is the one that will associate with C3b. Thus, we form a complex called as C3b, capital B small b, and this complex that is formed has enzymatic activity and can function as a C3 convertase. The C3 capital B small b, which also is the C3 convertase, can cleave more C3 and form more C3b that results in an amplification loop. Thus, we have more C3a fragments that function as anaphylatoxins and mediate inflammation, as well as C3b that coats the pathogen and helps in the elimination of the pathogen. To look at how C3a functions as an anaphylatoxin, we should remember that C3a is a soluble fragment and hence it can diffuse away. It is able to function as an anaphylatoxin by promoting acute inflammation. And it does that by increasing the permeability of blood vessels that results in proteins and immune cells being able to leave the blood and come into the surrounding tissues where the C3a is being made. It promotes the chemotaxis of the immune cells, thereby they know where to go to find the infection. It also activates some immune cells, and thus C3a is able to promote acute inflammation. The C3b that is formed has a reactive thioester group that is able to bind to hydroxyl or amino groups present on proteins or other molecules on the cell surface of the pathogen. And thus the surface of the pathogen is coated by C3b, which also functions as an opsonin. It is able to function as an opsonin because immune cells have receptors that can recognize C3b, and thus they bind to C3b that is present on the surface of the pathogen, effectively mediating the phagocytosis of the pathogen, which is also called as opsonization. Thus, the immune cells are able to opsonize the pathogen and destroy it, resulting in elimination of the pathogen. In addition to functioning as an opsonin, C3b can also bind to the C3 convertase that was formed before, which is C3b, capital B, small b, and it results in the formation of the complex C3b2, capital B, small b, which has two C3b molecules and one capital B, small b molecule. This complex functions as a C5 convertase, and thus it is able to interact with C5 convertase and cleave that C5 convertase into two fragments. The C5a fragment that is formed functions as an anaphylatoxin, while the C5b fragment that is formed can activate the terminal stages of the complement pathway that results in the formation of a membrane attack complex. Once C5b is generated, it is able to bind to another complement protein called C6. The complex of C5b6 is able to recruit another complement protein called C7, and the interaction of C7 with C5b6 is able to expose a hydrophobic region on C7, and this allows the C5b6-7 complex to be on the plasma membrane of the pathogen. They recruit another complement protein called C8, and thus we form the complex of C5b, 6, 7, and 8. This complex can recruit another complement protein called C9 to form another complex. But the neat thing is it doesn't recruit only one C9 molecule. It can recruit many C9 molecules, anywhere from 10 to 16 of them. And these C9 molecules arrange themselves in a ring structure to form the membrane attack complex. The membrane attack complex forms a channel-like structure in that phospholipid bilayer of the pathogen. And this pore-like structure is able to disrupt the proton gradient across the membrane of the cell because it allows the flow of different ions and other substances between the cell and the environment. The membrane attack complex also allows water and enzymes like lysozyme to enter the cell from the environment, and thus the cellular integrity is compromised, which results in the death of that cell. Now let us look at the second way by which the alternative pathway can be activated. And for this, we have to go back to the complement protein C3. Now C3 has a thioester that is present as part of its structure that can undergo spontaneous hydrolysis. And this is called as tickover. So when water reacts with C3, then this hydrated C3 undergoes hydrolysis and is denoted as C3H2O. This hydrolyzed C3 can now bind to factor B. And by binding to factor B, it facilitates an interaction with factor D that can cleave factor B that was bound to that hydrolyzed C3 or the C3H2O. This results in the cleavage of factor B to form two fragments, which are BA and BB. Now the BB fragment associates with the hydrolyzed C3 and forms the complex C3H2O-BB, which is also called as the fluid phase C3 convertase because it has enzymatic activity. So this fluid phase C3 convertase will interact with C3 molecules, and it can cleave them to form C3A, which is an anaphylatoxin, and it is soluble and hence diffusible. And it creates the other larger fragment called C3b, which can now bind to the surface of the pathogen. Now this C3b that is bound to the surface of the pathogen can interact with other factor B molecules. And when that happens, then this factor B that is bound to C3b can now interact with factor D that will cleave the B protein to give you capital B small b. And we already know that C3b that is bound to capital B small b is also a C3 convertase. And this way we get more and more C3b that is made. With this, we come to the end of the alternative pathway, which we saw can be activated in two ways. The first way is through the activation of the classical or lectin pathway, which results in the generation of the fragment C3b. The C3b fragment is able to interact with another complement protein called factor B. The C3b factor B complex is able to interact with another complement protein called factor D, which then cleaves factor B into two fragments called BA and BB. The BB fragment remains associated with C3b, and this complex has enzymatic activity and is called as the C3 convertase. This C3 convertase will cleave other C3 molecules to give rise to C3A and C3B fragments. C3A is an anaphylatoxin and can induce inflammation, while C3B is able to covalently bind to the surface of the pathogen and thereby coat the pathogen. C3B also functions as an opsonin and mediates the phagocytosis of pathogen that is coated with C3B. Additionally, the C3b can associate with the C3b-BB complex to give rise to the C3b2-BB complex. And this complex functions as a C5 convertase, which will cleave the complement C5 protein into C5a and C5b fragments. The C5a fragment is a potent anaphylatoxin, while the C5b fragment that is formed can initiate the formation of the membrane attack complex by recruiting other complement proteins like C6, C7, C8, and different molecules of C9. Another way that the alternative pathway can be activated is through the spontaneous pathway. In the spontaneous pathway, the complement protein C3 is able to be hydrolyzed spontaneously by reacting with water. This interaction with water mediates the hydrolysis of the Thioester bond that is present in C3, and it is denoted as C3H2O. This C3H2O is able to interact with factor B to form the complex C3H2OB. This complex is able to interact with factor D, which will then cleave factor B into BA and BB. BB remains associated with C3H2OB and is also called as the fluid phase C3 convertase. This C3 convertase can then cleave C3 to give rise to C3b and C3a. And this C3b that is formed can then associate with factor B, as we've seen in the past, which results in the formation of ultimately the C3b-BB, which is also a C3 convertase. And thus we are seeing more and more C3b being made through the activation of these pathways. This C3 convertase will then generate more C3b and ultimately give rise to a C5 convertase. Through the action of the C5 convertase, C5b will be generated, which will then result in the formation of the membrane attack complex. So this is how the alternative pathway of complement activation works. With this, we come to the end of the talk where we learned about the activation of the alternative pathway through a spontaneous process and through the classical and lectin pathway. We also looked at the outcomes of the activation of the alternative pathway, which results in the elimination of the pathogen as well as the induction of inflammation.